Hello, welcome to MLS QR. Today we're going to have a quick review of red blood cell enzymopathies, glucose 6 phosphate dehydrogenase enzyme, and also how it fits with the hexose monophosphate shunt. And also, we're going to talk about pyruvate kinase deficiencies. Red blood cells are remarkably tough and resilient single cell transporters, playing a vital role in our bodies in so many beneficial ways to maintain health and homeostasis. But these tough and resilient red blood cells are also vulnerable to genetic enzyme defects called red blood cell enzymopathies. Let's focus on two important ones found in our ACP study guide: glucose 6 phosphate dehydrogenase and pyruvate kinase enzyme deficiencies. Red blood cells need these functional enzymes to maintain glycolysis, preserve their biconcave shape, elasticity, and deformability of their cell membranes. Iron must be kept in the reduced 2 plus valence ferrous state in order to carry oxygen and convert ferric iron from its 3 plus valence state in methemoglobin, which cannot carry oxygen, back to its functional ferrous iron. A strong defense against damaging oxidative molecules like hydrogen peroxide and oxygen free radicals must be maintained to protect hemoglobin from denaturation and other biomolecules from degradation. To survive 120 days in circulation, red blood cells need to metabolize glucose. Since mature red blood cells do not have nuclei and mitochondria like their erythroid precursors, they need outside energy sources, and they rely upon anaerobic metabolism of glucose in glycolysis. As a living, breathing tissue, a unit of packed red blood cells need the added sugars in the preservatives adsol to survive 42 days in the blood bank and CPDA1 to survive 35 days before the unit must be discarded due to reduced viability of the red blood cells. For a quick background in biochemistry, familiarize yourself with glycolysis or the Embden-Meyerhof pathway. The hexose monophosphate shunt branches from glucose 6 phosphate of glycolysis. The glucose 6 phosphate dehydrogenase enzyme is part of the hexose monophosphate shunt, which branches off glycolysis. To protect against oxidative stress from hydrogen peroxide and reactive oxygen free radicals, red blood cells need reducing molecules NADPH, reduced glutathione, and the critical enzyme glucose 6 phosphate dehydrogenase and the hexose monophosphate shunt. As you can see here, glutathione peroxidase will use the reduced glutathione to neutralize and reduce hydrogen peroxide to harmless water. These enzyme reactions are all linked together, but let's simplify in the next slide. G6PD is an important enzyme as part of the hexose monophosphate shunt, a branch of glycolysis, which is used to generate reducing compounds like reduced glutathione and NADPH, which will neutralize reactive oxygen species and hydrogen peroxide and convert them to harmless water. When there's a deficiency of G6PD, the hexose monophosphate shunt shuts down. So we see the benefits of the hexose monophosphate shunt for the red cells. The enzyme G6PD generates NADPH for glutathione reduction. Here are some interesting facts about G6PD deficiency. Antimalarial drugs such as quinolones and other drugs may cause hemolysis in susceptible individuals with G6PD deficiency due to the oxidative stress on their red cells. Can you predict what initial lab works that will help the clinician diagnose a general hemolytic episode? What lab work will be more specific for G6PD deficiency diagnosis? What do you expect to see in the right Giemsa stain on the peripheral blood smear of a patient with G6PD deficiency? Bite cells are also called degmacytes. They're related to blister cells, helmet cells, schistocytes, the fragments of red blood cells, and these are commonly seen in hemolytic anemias, also thrombotic thrombocytopenic purpura, and disseminated intravascular coagulation, all signs of hemolysis. Did you notice I didn't mention Heinz bodies? And we need to do a different stain, a supravital stain, and we'll try it with the new methylene blue stain. New methylene blue is a supravital stain used for reticulocyte count, and you can see Heinz bodies using the new methylene blue, and these are denatured hemoglobin precipitates. And notice that you also see the bite cells. A more specific diagnostic test is the G6PD activity test. G6PD activity assay is also called the fluorescent spot test. The most specific test for G6PD deficiency is the DNA-based mutation test, DNA testing. Pyruvate kinase deficiency is the second most common enzyme deficiency. And do you know where it is in glycolysis, the pyruvate kinase enzyme? In general, when there is an enzyme deficiency, the products are deficient and you have substrates backing up, and both may cause problems with the pathophysiology. What do you expect to see in the peripheral blood smear of a patient with PK deficiency? They resemble echinocytes with the sea urchin spikes. And because of the pyruvate kinase deficiency, there's an inability to synthesize pyruvate and ATP energy molecules, and they're unable to replace important enzymes necessary for things like the sodium potassium ATPase pump, which requires energy, and they lose their ability to maintain their shape, and they have dehydration, shrinkage, and hence the echinocyte shape. Burst cells are echinocytes, both nonspecific signs of PK deficiency, but most commonly you'll see burst cells when there is a traumatic tap with a 21 gauge needle, and then you'll see hemolysis in the chemistry specimen. Likewise, the fluorescent spot test for pyruvate kinase deficiency is a qualitative test used for screening. The DNA molecular test for pyruvate kinase gene mutations is superior in sensitivity and specificity. It's been a tough subject with a lot of biochemistry, but let's see if we're ready for some Q&A. And these are based on the ASCP study guide. And they're similar, but not exactly the same. Thank you for your time, and